Good evening, my dear students. Good evening, Vasa. Good evening, Dhanalakshmi. Today, we are going to discuss about the...
Good evening, sir.
Good evening, Paul. The portion is called as mutation, continuity of the molecular basis of inheritance. Just note it down. This is very, very important for mutations. What is meant by mutation? Mutation is nothing but the sudden changes. Okay, sudden changes which occurs in the gene or nucleotide in a particular chromosomes. So as a result, what happened, there is a variations occurs in the progeny. Okay, it may cause many factors. Many factors is there, physical, chemical, and biological factors also induce the mutation. Is it noted on? This is very, very important. What is by mutation? Mutation is a sudden heritable modification in an organism's triads. This triads is nothing but what is that? Nucleotide or DNA or genetic code. Okay. So as a result, what happened? There is a variations occurs. Sometimes it may occurs in the naturally. Okay. During the meiosis. During meiosis, crossing over also takes place. That time, the mutations also occur. Likewise, some of the changes takes place with the help of the human being, otherwise called as artificially. Okay. Also, some mutations also takes place in commercial purpose. Okay. Some of them are beneficial, some of them are non-beneficials. Okay. See here. Genetic variation that forms, already told you, genetic variation. How it becomes variation? Variation comes by the process called as mutation. Okay, that form the basis of the differences between the related organism as well as the diversity of among organisms, or assigned to a process called as, what is that? Mutations. Already you know very about some of the organism already present, isn't it? That animals, take an example, so 100 years before, some of the animals also there, same animal also now are living in the world, isn't it? Compare with these two organisms, it is not similar. It may be slight variation or some of the changes occurs within the gene. Okay, take an example of human being, already you studied it. Okay, Neolithic, okay, Mesolithic, and so on and so. And also the Homo sapien, Homo erectus. Okay, likewise. So what are the changes? There is a traditional changes also occurs, isn't it? With the help of the gene, mutation. Take an example, tail, already present in it. They are all are completely not utilized. They are all are considered in vestigial organ. Okay, so just imagine there is a variation occur in the particular organism due to the gene. Okay, see that one. Next one. The term mutation, just to note it down, my dear student. This is very, very important. The term mutation was first coined in 1901 by Hugo de Vries. Okay, the term mutation was first given by in 1901 by Hugo de Vries to explain the variations he observed in the plant evening primrose. Don't forget, my dear. The botanical name is called as Enothera lamarckiana. Don't forget the term mutation. Who is that? Which year? And which plant? Primrose. Primrose. Okay. So this is also called as, what is that? Botanical name? The Enothera lamarckiana. Sometimes they may ask the questions. The term mutation was observed by the which one of the following plants? So you have to mention Enothera lamarckiana. Okay. For the next one. So he is a scientist. So Hugo de Vries. Who was Hugo de Vries? This is the plant, evening primrose. He was a Dutch botanist and one of the first geneticists. Okay. His period See here, 1848 to 1930. These are not known, no need. But you need which one, the scientist name, which plant, and which country. Sometimes it may be very rare, but note it down. It's also known mainly for suggesting the concept of, see, these are the points have been reported by the scientist Hugo de Veris. First one is genes, already you know about. Suggestion of the concept of the genes. Rediscovering of the law of heredity in the year 1890, while unaware of George Mendel work. Before Mendel, he also stated the statement regarding heredity, law of heredity. Okay. Unaware of George Mendel works. See, he do not know. He doesn't know about the Mendel's work. Before that, he stated the statement. Okay. And then introducing the term mutation, already told you in the intro part. Developing a mutation theory of evolution. This is also very, very, but here they may ask the question, two things is there. Who unaware of the George Mendel work? Like that they may ask the question. The term mutation was introduced by the name of the scientist. They may ask the question. And mutation theory of evolution. This is also another question. Maybe ask the question about regarding Hugo de Veris. Okay, are you clear in this part? Idea. Listen. The mutation is the basic importance of genetic, and it is defined as a sudden discrete changes, otherwise called as inheritable changes in the genetic material. What is the meaning of genetic material? Genetic material is nothing but the gene or DNA or nucleotide, whatever it may be. But at the same time, which is heritable. They may be transferred from the one generation to another generation. Which one is transfer the character from the one generation to another generation means DNA only, already you know well about it. That's why we are called DNA as a genetic material. Characters, skin color, eye color, okay, and also diseases, whatever it may, resistant or dominant diseases also there. Blood group, multiple alleles, okay, alkaptonuria, whatever it may be, and behaviors, okay. So these are all are related to the genes, genetic materials. Genetic materials, nothing but the DNA. But gene is also made up of which one, nucleotide. So that is very, very important one. So according to these things, they have explained another part. Causes of mutation, how it will be taken. Mutation, how it will be occur. So what is meant by cancer, my dear students? What is meant by cancer? Dhanalakshmi, what is meant by cancer? Varsha?
It is caused by the tumors, sir.
It is caused by the tumor. No.
Yes, sir.
Tumor is nothing but the, what is that? It's one of the mass of cells only, isn't it? Varsha? Yeah, Varsha.
Yes,
Yeah. Oh. What is my mutation? Cancer. Cancer is also
cancer occurs because of oncogenes.
very good oncogenes. So what happened inside the Varsha? Inside, what happened? Why it is called as oncogene? Why it is called as tumor? There also.
Sir, like the cells, they lose the capacity of contact inhibition and they divide in excess. That is why the cells like tumor occurs. And because of the detection of tumor through MRI scan or CT scan, we can get to know that a person is affected with cancer.
Okay, very good. But you are telling about that all those things. Main point, what is that meaning? Main, what happened inside the cell? Just you know very well about the cabbage, isn't it? Both of you, you know very well about the cabbage. Cabbage inside what they are having in each and every layer is there, isn't it? Likewise, each and every cell undergoes the mutation. Then only the multiplication takes place. The multiplication cells converted into neoplastic cell. That means the normal cell converted into neoplastic cell. The neoplastic cell converted into cancer. The cancer may be tumor or connected with immunovariable, but there are 4 types is there. Carcinoma, sarcoma, lymphoma, and multiple myeloma. Okay, it started from the outer to inner. Already we discussed in previous classes. Okay, anyhow. But causes of mutations, the spontaneous mutation is there. Spontaneously occurs. How it will be? The mutation occur randomly, naturally, and automatically die to the internal reason without any relation to any external factor. Automatically it happen inside the body. Sometimes what happen, spontaneously occurs. Such type of mutation is called a spontaneous mutation, otherwise called as background mutations. Okay, the copy error. This mutation involve incorporation of the wrong base during DNA replication. This is very, very important. The spontaneous mutation occurs, right? Okay. How it will be occurs? Randomly, naturally, and automatically occurs. That's why it is called a spontaneous mutation, right? But at the same time, this mutation involve incorporation of a wrong bases. Incorporation means A, T, right? But it oppositely, where 149 gene is A, adenine and guanine, it may be replaced by guanine and cytosine during replications. So this is a error. Copy of our last class we were studied. DNA replication. What is it? Exact copy. Adenine, thymine, guanine, cytosine. Likewise, okay. Suppose if it is changed in the position of the locus. Locus means suppose 10th position, 11th position, 12th position. Imagine like that, 10, 11, 12, A, T, G. So corresponding gamete is also same, A means U. T means T, it's not coming, U. U means, about A. G means C. Suppose if it is replaced by some other changes, what happened? It lead to mutation. When it occurs, during DNA replication process, wrong bases also incorporated, joined together, as a result, the mutation occurs. It is naturally occur. Okay, this is the main one. See that? So maximum, sometimes they may ask the question, this type one also. Which of the following correctly differentiate between the spontaneous and also induced mutations? Listen here. The 2 point has been given here. The induced mutations are caused by error in natural biological process, whereas spontaneous mutations are caused by the mutagenic agent in the environment. Spontaneous already we studied. What is that? Any factors not involved. Automatically it occurs inside the body only. See, second point. The spontaneous mutation are caused by error in the natural biological process. That is very, very bad. Whereas induced mutations are caused by the mutagenic agent in the environment. This is the opposite of this one, isn't it? So this is the correct way. See here. So this is the DNA structures. These are the genes. How it happens? See that the genetic material. DNA is a genetic material, isn't it? The spontaneous means what happened, occur randomly due to the error in the natural biological process. Induced mean caused by the mutagens. Mutagens is nothing but what are the mutagens here? Itolavtrates, X-rays, pollutants, toxin, and chemicals. These are the outer sources from the environment. It induces the mutations. Okay, such type of mutations are called as what is that? Induced mutations. Induced by the chemical substances. That's why it is called as induced mutations. Spontaneously, naturally occurs. Such type of mutation is called as what is that? Spontaneous mutation. See, the induced mutation, these mutations are produced in the response to specific external factor and chemicals. Now only we start that one. Okay. Muller, don't forget my dear students. Muller, 1927, was the first to produce induced mutation. Don't forget. Take notes. In Drosophila melanogaster by exposing them to X-rays. Already you know about. Okay, ultraviolet rays and X-rays continuously fall on your body. What will happen? It will lead to mutate to the cell. As a result, you will get skin cancer. For example, suppose if it is outermost. Suppose if it is inside, take an example. Those who are taking X-ray in hospital, that particular person, per day 100 X-rays or 200 X-rays, sometimes in big hospital, about 500, 300, 1,000 it may be lead to, morning up to evening. So what happened? That fellow, they're wearing, which one? The lead. Lead, it is made up, the coat-like structure is there, it is made up of lead, because it is not easily penetrate the lead. Okay. So that person, suppose if it is not wearing, if he doesn't wear the coat, what will happen? It will be fallen to cancer within short period, or 6 months or 10 months or 1 year, even 5 years sometimes, depends upon the immunity. So likewise, the Muller also analyzed one of the animals, Drosophila melanogastrus, in the year 1927, by exposing them to X-rays. The specific factor and chemicals of the environment that induce the mutations are called as mutagens. Which substances they can induce the mutagens? That substances are collectively called as what is that? Mutagens. Suppose if it is virus, what is it? Oncogenic viruses. Okay, they also consider getting mutagens only, but it is what is that? Microbials, animal sources, living sources. Okay, ultraviolet is not living, X-ray is not living. Another chemical substance, take an example of dust particles. They are all comes under getting inorganic substances, organic substances. Okay, organism means microorganisms. Okay, virus. Okay. Point mutations. What is the meaning of point? You know about each and every DNA molecule is made up of bases, nitrogen bases. Any particular point, the particular base is replaced by or insertion, deletion, addition, substitution. So many processes there that they are all considered the chromosomal aberration. Okay, just to go and check the Google, what are the changes which occurs? Already we discussed, isn't it? Shift mutation, addition, deletion. Okay. In particular point only, the particular base only will removed. See that? Base substitution and insertion or deletion of the one nucleotide are known as point mutations, because they occur at a single point on a chromosome, single point. Take an example, a particular nucleotide is there, 50 nucleotide in a gene sequences, Take an example of 15th one. Okay. Suppose G is there, G, A, G, like that. It is replaced by, what is that? Another one, ACC or AAA, like that, that is, what is that? Substitution. Insertion means that point you are inserting. Inserting means any other 3 nucleotide or codons are inserted that particular point. Deletion means the particular point will be completely removed from the nucleotide base. As a result, what happens? There is a mutation in particular point. Okay, so this is a very, very important. Base substitution, replacement. Okay, about the substitution. Take an example, say any one of the sports, okay, cricket or whatever it may be, football or basketball, whatever it may be. 10th one, please substitute 11 or 12, like that. So likewise, the particular base will be substituted or replacement by the another one. It is also lead to, what is that? Changes or mutation. See that? Base substitution is the replacement of the one base by another. Example. One base by another. Example. If your DNA molecules usually contain guanine at a certain position, take an example, 15th position, already told you. It can be replaced by, which one But adenine takes the place of guanine. So the guanine is replaced by adenine. What happened? There is a change in the 15th position. As a result, the mutation is also takes place. Such type of, what is that? Introductions or additions is called as substitutions. See that? Then the base of the substitution has occurred. There are 2 types of base substitutions. What are they? See that? Transition and transversion. Transition. What is the meaning of transition? See that these involve the replacement of the purine with another purine. What is by purine? Adenine replaced by guanine. Okay. Of a pyrimidine with another pyrimidine. What is the meaning of pyrimidine? Innoverable. Cytosine replaced by thymine. Okay. That is the things. Okay. See that transversion. These involve the replacement of the purine with the pyrimidine or vice versa, whatever it may be. Suppose in particular place, the adenine is there. It can be removed and also replaced by cytosine. Purine removed and also addition of pyrimidine. Suppose and that place the pyrimidine is present. Remove the pyrimidine and add the purine. This is transversion. These are the two types of substitution present in it. See that one? You can easily identify. This one is the normal sequences. See here. See the sequences here. C, A, T, T. The fourth place there is a T is there, isn't it? Thymine is there. The thymine, what is that? Automatically join with the adenine, double bond already. Thymine always adenine. Thymine, adenine. Here thymine is there, here adenine is there. This is the normal sequences. Transversion. See that transversion, what is the meaning of transversion? T is replaced by A. TA is replaced by GC. This is the adenine is the purine molecule, thymine is the pyrimidine molecule. Here guanine is the adenine, purine molecule, cytosine is the pyrimidine molecule. This is replaced in this position. This is the fourth place. 1, 2, 3, fourth place replaced. Okay. See that T and A is here, but here replaced by G and C. So the sequence is completely what happened in changed or mutated. So this is called as transversions. Suppose it may be changed another one also. See, place it here C also and also again G also. You can invertedly also replace it, substitute it. It is also changes. Okay. So this is a one type, transversions. So another one is, see here, so many combinations you can transversion takes place. Adenine always combined with the molecule of thymine. Okay, these are the two things is there. Likewise, cytosine and guanine. Transversion, you can replace it any one, any direction, thymine to cytosine, cytosine, thymine, or adenine to guanine, guanine to adenine, guanine to cytosine, whatever it may be, you can replace it any directions. So they are all comes under the heading is known as what is a transversion. So as a result, what happened? The nucleotide, the sequence also changed. That is mutation. Okay, see that one. Frame shift mutation. Another one is called as frame shift mutation. What is the meaning of frame shift? Particular gene, for example, 40 to 50. Okay, remove completely 40 to 50, remove it and add another 40 to 15. Another sequences also added. That is called insertion. Deletion means what is that? A particular 40 to 50 completely remove it. Okay, 1 to 40. Then start with the next one, 51. In between 40 to 41 to 50, completely lack. So these are all comes under the headings known as frame shift mutation. See that? Frame shift mutation changes the reading frame of your genes. There are two types of frame shift mutation. One is called as insertion, another one is called as deletion. Insertion means already you know additions. You are adding 41 to 50. There is a space is there. You have to add the 41th place. What is that? Any other 10 nucleotide, any other genes inserted in 41A, 41B, 41C like that up to 10. Okay, see insertions. As the name implies, this involves the insertion of the one or more extra nuclei. One or more means I told it 10. Here one is also inserted the particular nucleotide. What is that? It is also insertion. 5 or 3 or whatever it may be. It may be up to your business requirement. Extra nucleotides into a DNA chain. This is very, very important. That is insertion. Deletion means remove completely. See that this result from the loss or one or more nucleotides from your DNA chain. So this is the another important. These are the two process occurs in the frame shift mutation. Frame means completely remove, completely add. See that? You can see that structure. You can easily identify. See that one? So this is the original insertion. Okay. This is the original place. See, C, T, G, and G also there. After the G, what happened? What happened? You are adding TGG. TGG completely the shift will be added. Okay. See, then next one is A and G as it is. This is the what? 1, 2, 3, 4. 5 and 6 and 7. Sorry, you are adding, isn't it? Normally 2, 4, 6 only is there. Here, how many is there? 6 plus 3, 9% in it. Okay. So this is very, very important. This is called as a frame shift mutations. Go to the next one. This is also deletion. What is the meaning of deletion? See that this is the original sequence is there. See, 1, 2, 3, 4, 5, 6, isn't it? T, A, and C, and T is there. What about here? The T is completely removed from this one. This is called as frame shift. Otherwise called as deletion. Okay. Sometimes it may be completely removed of these sequences also. Okay. This is also another type. See the substitutions. This is the normal place. Okay. So where there is a dark black color they have given, see that here G and C only is the third position, isn't it? But here what happened, substitution. Remove these two nucleotide, or they are adding A and T. So in this part, what happened? Completely remove the particular part. This one is completely removed. This third place is removed here. Here insertion means addition of one more is there in this part. This is the mutation process. Substitution. Substitution means replacement. Okay. Deletion means remove from the chain. Insertion means addition of one more nucleotide in the chain. Okay. So do you understand this point, my dear? Or you follow me?
Yes, sir.
Okay. Any doubt, my dear? Versa?
No.
Versa, what about Versa?
No, sir.
Okay, go ahead. See that one. Another is also one. This one. See that methionine, lysine, AEG is methionine, lysine, phenylalanine, glycine, aniline. These are the normal one. Each three is encoded by a amino acids. Here what happened, See that one? Only one will be changed. That is called as nucleotide substitution. As a result, the particular amino acid completely changed. Here the glycine is mentioned. But what about here? See that? GGG. Okay, GGC here. Here what happened? AGC. So the A is substituted here. As a result, what happened? That amino acid sequence also changed. That is serine. Whatever nucleotide deletion. Deletion means what is that? See here in this part. So U is, what is that? Deleted here. As a result, what happened? It is also added. This U is removed means join with UU and G like this. See, UU, G's. So as a result, the amino acid completely changes, isn't it? This completely, these three nucleotide also, or codon also completely changed. Okay. Suppose if you are inserted, what happened? This is also a changing. So these are all are induced, what is that? Mutations. Okay. And then missense mutation. Another one is very, very important, my dear students. Sometimes they may ask the questions, missense mutation. What is missense mutations? Change of only one codon may be missense when it is changed the insertion of one amino acid. This is the missense. Already we saw it in the previous slide, isn't it? Only one codon. Don't forget, my dear, one codon. One codon means each and every amino acid is encoded by 3 codon. AUG, methionine. Okay. Particularly A is removed, that is one codon, A, adenine. Okay. So, adenine is removed, you add some other one, U or T or G or whatever it may be. What happened? No, the amino acid sequence will be changed. That is called as missense mutations. This may or may not change the properties of the polypeptide. Sometimes it may be changes take place. Sometimes what happens? There is no any remarkable changes which occurs within the nucleotide. Okay. So for example, sickle cell anemia is caused by a change in the chain of hemoglobin. Already we know about. At the 6th position, where the amino acid glutamic acid is replaced by valine. Already we saw it in the chromosomal aberration. Okay, recessive gene inheritance and sex-linked inheritance. Okay, glutamic acid, which position? 6th position. Where? In the sickle cell anemia of the hemoglobin. Hemoglobin, where it present? RBC. Hemoglobin, you know about 2 alpha chain, 2 beta chain is also there. Okay, the 6th position of the amino acids, which one? You know, the glutamic acid is replaced by valine. See that one? Structure. See? So these are the point mutations. Silent mutation is also there. You know, no mutation. In this category, no mutations. See the TTC, AAG. So this is mentioned as a lysine. Okay. So this one is called a silent. Silent means lysine. This is also another type. Nonsense. This is stop codon. When it comes UAG, what happened? It is a stop codon. Okay. Missense. Conservative and non-conservative. These are arginine and threonine. These are all our point mutation. Particular gene is also replaced by the. So as a result, what happened? The mutation takes place. Okay. So go to the next one. The nonsense. Nonsense mutation. This one, missense. This one's nonsense. So what is the meaning of nonsense? Means, see here, in this mutation, in sense codon is changes to stop codon. That is called as termination codon, otherwise called as stop codon. Okay, they are called as what is that? Nonsense codon, because they will not indicate any amino acids. Just go and check it. Stop codon. They will not indicate any amino acids code. Okay. So previous also we saw it. One is stop codon. See that this one is stop codon. It's UAG. See, others are, they mention the amino acid sequence. Lysine, arginine, threonine. See here. AAG, lysine. AAA, lysine. AGG, arginine. ACG, threonine. Whatever here, they didn't mention the, what is that? Enzyme, sorry, amino acids, isn't it? Such type of codon is called as nonsense codon. They cannot be able to mention. They cannot mention the amino acid. Such type of codon is stop codon, at the same time termination codon. Okay. See here. Another one is silent mutations. Silent mutation takes place with the help of the same sense mutations. Silent mutation does not cause any changes in the expression of the gene. It occurs, changes occurs, but it do not express the gene. Okay. Such type of changes is called as, what is that? Silent mutation. Nonsense, silent mutations. Okay. See that one? So these are original sequences, base substitutions, base insertion, base deletion. Already we saw it, these things. Silent mutation. Same sense mutations. Silent mutation does not cause any changes in the expression of the genes. Already I told you. This is original, base substitution, and base insertion, base deletion. Okay, these are, see that only one point is removed here. Addition or removed, whatever it may be. You can imagine. These are all silent mutations. Base pair substitution can create a, see here, missense mutation. Okay, see, DNA sequence is this one, GAA. Okay, correspondingly, it connected with the CTT, guanine, cytosine, adenine, thymine. It may be, what do you have? What happened? Change in the second place. See here, second place. G, TA, C, AT. See here, the second position of the codon will be changed. Okay. mRNA, GAA is the normal one. See the second one, GUA. Protein, so GLU. So glutamic acid converted into valine. It's nothing but the amino acid, because amino acids are protein building component. Okay, so that is the reason. So proteins is nothing but the amino acid. Just imagine here, the glutamic acid replaced by the valine, because the changes is where the center places. Likewise, nonsense also they have mentioned DNA, TTA, so corresponding AAT. So reverse mill. This is called as polyandrome. See that ATT, A, sorry, this one is not there. Here what they replace here, the second person. See here, A also replaced by T. It may be reversely. Okay. mRNA, UUA, here UAA. Leucine is also here, the stop codon. UAA is also one of your stop codon. Silent mutation, just imagine the last place will be replaced here. Triple C, triple G. See here the last one, CCA. Here the last one is replaced. Here GG, T only given. So mRNA, what happened? Corresponding. So the CCA only you will get. So the proline, here the proline. So this is silent mutation. The same amino acids replaced. The amino acids, there is no change, but codon also changed here. Okay. The protein is here. Here also the, proline is also here. Proline also here. But what happened, the gene codon is also, the single codon is also changed here, isn't it? So, but what about the amino acid sequence? Amino acid sequence, there is no change. Proline and so proline. So this type of mutation is called a silent mutations, my dear. Don't forget, this is very, very important. Sometimes they may ask the question, silent mutation, give an example, means proline and proline. Okay. So triple C, mention proline. CCA, proline. Okay. Leucine, glutamic acid, valine, already in our, but this one is called as, what is that? VAR in sickle cell anemia. Okay. Go to the next one. Question. Okay, question part here. I have given 5 questions. It's a very, very important, my dear students. Will you please tell me question number 1? Read the following statement and choose the set of correct statements. Euchromatin is loosely packed chromatin. Heterochromatin is transcriptionally active. Histone octamer is wrapped by negatively charged DNA in nucleosomes. Histones are rich lysine and arginine. A typical nucleosome contains 400 base pair of the DNA helix to 50. This part, which one is correct one, my dear? Choose the correct answer from the options given below. Varsa and Dhanalakshmi. Already we saw it. Yeah.
So option 3.
Option 3. Is it correct? Varsa?
Yes, sir.
Oh, okay. What about Dhanalakshmi?
Same, sir.
Same. Arisaya?
Yes, sir.
Okay, we will see that one. So answer 3 is the correct one. How it be, the solution is that heterochromatin is transcriptionally inactive in nature. First point. So here what they have given? Inactive. Here active has given, isn't it? So the third point, what is it? A, C, D only correct. Okay. So B and A, what is that? E, it will not come. So what are the points? G, C here, the heterochromatin is transcriptionally inactive. A typical nucleosome contains 200 base pair of the DNA helix only. Okay, here they have given 400, isn't it? Okay, so what about the next one? The euchromatin is loosely packed chromatin region. Is it correct or not? Correct. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called as nucleosome. It is also correct. Histone are rich in basic amino acid residue lysine and arginine. Other parts also correct. Very good. Both of you correct only. So the answer is A and C and D only. See that euchromatin is loosely packed chromatin. And C, the histone octamer is wrapped by negatively charged DNA in nucleosomes. It is not come because it's inactive nature. Histones are rich in lysine arginine. It also comes. And typical nucleosome contain 200 base pair, not the 400 base pair. Okay. Okay. Go to the next one. See that? This one. Give me the answer, exact answer, which one is correct. So solution also given here. Okay. In the following palindromic base sequences of the DNA, which one can be cut easily by particular restriction enzyme? All are similar in nature. See that? 5 does, 3 does, 3 does, 5 does. See here, the first is C is the corresponding joining. Which one is the C? G, C. Always guanine combined with the molecules of cytosine. Thymine, adenine. Adenine, thymine. Thymine, adenine. Thymine, adenine. So guanine is cytosine and guanine, isn't it? But in this part, they have given the answer is 3. Which one is correct? No, GAATTC. Okay, this is the correct one. Why? They asked the question palindromic. Which one is correctly matched here? See that last one. When you are reading the 5' to 3' and 3' to 5', isn't it? Both are equal in measure. Okay, 5' to 3'. Here the 5' to 3', both are same. Take an examples, Malayalam. Spelling already in oral, MALAYALAM. When you are reading the left to right, it is same, and right to left also same. That is palindromic, isn't it? So which one is correctly matched here? See that GAATTC. See here, GAATTC. So this is the correct answer. Okay, are you clear, ba? Dhanalakshmi,
Sir. Yes, sir.
are you clear, Varsa?
Yes, sir.
Any doubt in this part, my dear?
No doubt, sir.
Okay, are you clear in this part? So this is very, very important one because G, AA and TTC is there. So you have to read 3' to 5' also similar structure. Such type of gene sequence is called as palindromic. Don't forget, my dear. And then third one. Given below are the two statements. Statement 1, restriction endonucleases recognize specific sequence to cut DNA known as palindromic nucleotide sequence. Is it correct?
Yes, that is correct.
Correct.
Yes, sir.
Yeah, right. Statement 2, restriction endonuclease cut the DNA stand a little away from the center of the palindromic site in the light of the above statement. So these two are correct only because already about restriction endonuclease, it cut any part, particularly palindromic position only. But at the same time, it will not cut both the stand exactly. It may cut like as what is a soap box, like it maybe give some space, right side or left side, whatever it may be. Okay. Then only, okay, the nucleotide will be separated. See that answer is both statement and 1 and 2 are correct. Example, okay. See that one, I will show you how it will be. See here. Take an examples. So this is a sequence of DNA molecule. Just imagine this one is called as 5' and 3', isn't it? Imagine 3'. Opposite, about this one is 5'. Okay, here already about 3'. The endonuclease, what is that? Endonuclease. Okay, where there is a cut, it will take place like this. Here there is so many, what is that? Sequences there, amino acid sequences already about this one. Correspondingly matched, isn't it? Already about, just I put the small diagram only, just to imagine. Okay, opposite also is the already about. Okay. Likewise. Okay, just imagine this part is A. Okay, this one is also thymine. So likewise, you have to fill it. Okay, where there is a cut made, suppose take an example, in this part there is a cut. Okay, so this portion and next one is what is that? This is the portion they made here. The cut is like this. Okay, this portion only because the polyandromic genes. How it will be? So the after the cutting, after you made a cut, how it will be the DNA sequences? Imagine. See, the structure will be like this, isn't it? See, the structure will be like this, isn't it? So this is the structure you will get. So this portion, it comes here. In this portion, it comes here. Okay. So what is that? This is the place. Okay, so I have to put it like this. Okay, so this area is palindromic. See the space is here. It will not cut exactly. For example, take an example, the cut made like this. It is not like this, isn't it? It not cut like this. It is not cut like this, isn't it? Exactly. It cut like a, what is a polyandropic gene where it's located, but at the same time, it's away from the genes. That is the things. This is the part. This is the cutting edges. Okay. So this is a very, very important. Suppose when you are fixing these two, you will get this entire structures. Okay. Okay. See that one. Yes. Okay. So the answer is, which one is correct? You know, the second one is the correct. Both statement 1 and 2 second are correct only, because restriction and will cut the DNA strand a little away from the center of the polyandropic gene. Already I told you how it will becomes already, so it isn't it. And we will pass on to the next one is, and fourth one. Solution also given here and deeply. Okay. So already I mentioned in the diagrammatic aspect. This is another part. See, anyhow, just note it down. If the length of the DNA molecule is 1.1 meters, what will be the approximate number of base pair? Okay, the answer is 2. They have given is the answer. First one is, see here, the option given here, A 6.6 into 10 to the power of 6 base pair, 3.3 into 10 to the power of 9 base pair, and 6.6 into 10 to the power of 9 base pair, and 3.3 into 10 to the power of 6 base pair. Already you know about the human genome. Okay, approximately how much you know? Maximum 3.3 down, isn't it, my dear student? 3.3. Where there is a point 3.3, you know, 2 and 4 only the answer option, isn't it? These two will not come, so it is omitted. First and third is omitted one. Okay, from these two one, already you know about 3.3 into 10 to the power of 9. 3.3 into 10 to the power of 6. Already you know about 10 nearest, which one is there? 9 only is there. So this is the correct answer exactly. So how it will be calculated? There is a formula has given. This is direct question. Okay. Particularly suppose they are asking like they ask the question like this, you know. Solution C here. The number of base pair into distance between the two consecutive base pair. Okay. Equal to length of the DNA molecules. They can be calculated. Some of them are here missing. I will tell you next classes in this part. Okay. This is the direct question. 3.3 into 10 to the power 9 base pairs normally present approximately in human genome. Okay. This is the fourth questions. Fifth questions. In Hardin-Weinberg equation, the frequency of heterozygous individual is represented by. Will you please tell me Hardin-Weinberg law? Both of you.
Sir, in a Mendelian population, the allelic frequency is always equal to 1 and it is a constant, sir.
I'm asking the formula, my dear.
So, P plus Q the whole square.
Okay. What will be the expansion?
So, P square plus 2PQ plus Q square.
Okay. Which one is homozygous? Which one is heterozygous? Which one is dominant? Which one is recessive? You don't know?
P homozygous.
Okay. Homozygous.
And Q heterozygous, sir.
No, what I'm asking, no, homozygous, dominant or recessive?
Homozygous dominant, sir.
Okay. What about the Q?
Heterozygous recessive.
No. See here, Varsa.
Yes, sir.
Do you know that any idea about Hardy-Weinberg equation?
Yes, sir, I know.
Yes. Will you please?
So P.
Yeah. Go ahead.
So P is homozygous dominant and Q is heterozygous dominant.
Heterozygous dominant. Are you
Yes.
sure? Are you sure? Okay, listen here, my dear. So in this part, I have to share another slide also. Then only you can easily identify it. See that one. So see, my dear. So what will be the equation? You know very well about p square minus or plus here plus q squared, isn't it? Is it correct or wrong? So this is the, see here, p squared plus q squared plus 2, sorry, wait a minute. So I will type, then only it will be better. P squared, okay, plus, sorry, q square equal to what is that? This is the formula, isn't it? This is the formula. This one we will finish it up to this level. See, the next one is what happened now? Sorry, this one is also wrong. Okay, sorry. I'll put 2 outside. Okay. See here. So what will be the result? You know, p squared plus, sorry, plus q squared plus 2pq. You have to put here, okay, what is that? P squared. See that? P squared what? Q squared what? 2p q what? So this one is, what is that? You know, already you know what about p squared is nothing but what is that? Homozygous dominant. Okay. So the next one is, what is that? Homozygous recessive. What is the meaning of heterozygous? Means both the character present in it. This one is homozygous dominant, this one is homozygous recessive is that. Here PQ, both are present in it. So this is called as, what is that? Heterozygous. Now are you clear now?
Yes,
Hello?
sir.
Yes, sir.
Okay. They asked the question, which one, you know? Heterozygous, sorry, the spelling is here, mistake, no, hetero, okay. Heterozygous dominant, okay. So the heterozygous dominant character only they asked. They represented heterozygous only, they asked. See the question paper, you can, are you clear in this part, my dear?
Yes, sir.
So this is equal to 1, okay, p plus q whole squared, that is equal to 1. So you have to mention p squared and q squared. P squared is a dominant character, but it's also homozygous. It's nothing but what is there, you know? 2p is present. See here, 2 plus p, that is equal to p squared. Likewise, you have to tell q plus q. Sorry. Okay. Q plus q is equal to q squared. What about here? 2p and 2q also there. So the 4 is there. But at the same, both the character present, the dominant character as well as the recessive character. But here, P is the dominance. That's why it expresses the dominant. Okay. What they ask the question, see here, listen carefully. This is very, very important, my dear. Okay. So it is a, see that one. That's why I asked the question. So we'll finish it up to this level. I think the time is also up. See that one. See this one. In Hardy-Weinberg equation, the frequency of heterozygous individual. Heterozygous, what is that here? Which one is heterozygous here? According to the law, frequency, p squared, it will not come. Okay. And 2PQ is possible. Already we saw it, that one, heterozygous. PQ is also possible, but according to their laws, 2PQ it comes. Here they have given only 1PQ. What about the last one, Q squared? This is a recessive one. See here, the solution B in Hardin-Weinberg equation, that is equal to homozygous dominant individual. That is called as P squared. And 2PQ, heterozygous individual. This is 2PQ. And homozygous recessive individual. This is called as, what is that? Q squared. So this is the correct one. Which one is the correct? They ask the question, read the question carefully, then only you will get the answer. Otherwise, you will answer wrongly. Okay. Heterozygous individual, they ask. The heterozygous means these three only comes. It will not come C. So dominant, they didn't ask. Heterozygous means both are correct present, P or Q. Both P and Q. How much is that? 2PQ. You just go through the, which one you know? The equation, then only will get the answer. You got it? So that's all. I think this part, the part 3 is finished, my dear students. So you go through it in mathematical aspects. So many questions are there, particularly genetics, molecular base of inheritance, as well as the principles of inheritance. These two questions, these two part only, they are asking max oriented questions. And one more is the respiration, and another one, dihybrid, monohybrid, like that, you know, and multiple alleles, genetic and the equation. Sometimes they may ask the questions. So be careful, my dear. So follow it and practice, keep on practice, then only will get the marks. Okay. You have to download all the question paper and then practice it every week. Take 5 questions. Okay. 2015 onwards, you have to print out. Okay. Weekly once, you just go through it, where there is question, it comes from which chapter, like that. Okay, each and every question just carefully read. So first 2015, what they asked, in which part is very important one they have given in the statistical aspects. The next year, which one, how many questions are there? Like that, you have to, what is that? Analyze. Okay, go through it. And one more is an NCERT book. You have to thoroughly read up to 5 times. 5 times only read. Black letters you have to underline. Okay, my dear, thank you for the participation, and we'll see the remaining on the next class, my dear student. Thank you. Good night. Thank you, Pam.